Thank you very much, Rini, and university invite us, give a talk, and showing what a wonderful country is, and what a wonderful people are, and wonderful history. So I learned really many things about Armenia, and I'm thinking mostly Armenian people are very proud of their country and people and their culture. So that I'm very thankful that I had experience to share things in Armenia. So today my talk is going to be changed. It was originally about alpha-synuclein, which is Parkinson's disease related, but I had some suggestion that maybe broad audience, this talk will be more have a common interesting. So hopefully through this talk, I will find the collaboration or students who is interested in what I'm doing, and they are into it, this kind of work. So my research is mainly focused on protein structure. My background is physics actually, so when I want to learn about biology, most easy way approach was through the structure, because structural biology they use many methods, X-ray, EM, which is developed by many physicists, and while they are developing, they use biological material to study about biological function. So same way, I started my learning process through the X-ray diffraction method. And the study, when I started the project, actually I didn't know anything about biology because I purely studied material science and physics. So I did not know what is bacteria, what is a cell. It was very difficult to approach. But one thing I know about X-ray diffraction, so that's how I studied. And first, my project was actually a very challenging project. The reason I chose the project because I have a very little experience with the protein. Actually, this protein, Huntington protein, which is I crystallized, has been tried. Many scientists, famous scientists, try to crystallize, and they haven't done any progress with obtain crystal structure. But since I have no knowledge about biology, I say this is going to be a very interesting project. The reason that I know about this protein, who is sitting here, and my life partner, he suggests actually, why don't you try this? So I say, okay. And when I bring the project to my boss, he said, really you want to try this? So I say, why not? And that's how I started this project. This project is Huntington protein, which is a very large protein, which is related to Huntington disease. Huntington disease is one of the follicle expansion disease, which is when follicle region is expanded, certain threshold length more than the length the disease develop. The disease is usually brain-specific region of brain neuron start dying. So that is very interesting. There is 9 known protein which is shares follicle expansion disease. So when this protein follicle region is expanded, they have all these proteins is related to neuronal death, neuronal cell death. So I think that was a very interesting project in sense which is related to neuronal cell death. And so I crystallized this protein and then another protein, ataxin 3, which is the same polyQ disease-related protein. Then from this structure, I was thinking how this polyQ expansion can be related to disease. And I find that there is a connection between polyQ expansion and proteasome dysfunction. And that is the first part of my talk. And second part is after I started working on neurodegenerative disease, and I found that there are many other diseases too. So one of the interesting projects is Alzheimer-related project, which is alpha peptide, which is amyloid peptide 40-42 generation, which is 40 is non-pathogenic, but 42 is pathogenic peptide. And I found that how these two protein peptide is generated and how can be this is biologically related. And also another AD project, which is FOE 3 and 4. FOE 4 is Alzheimer-related protein. I found that FOE 3, 4 is different binding affinity with the LDL receptor. And another project is alpha-synuclein project, which I suggest the first time give a talk, but today I cannot give, so I hope next time I will have another time discuss about this project. And then there is a sigma receptor, which is ALS related project. This is overall my interesting project, which is neurodegenerative related protein. This is a Huntington protein, which is I put the N-terminal MVP tag and C-terminal tag, and I crystallize external 1 with 17Q. The Huntington protein has a very interesting sequence, which is 17 amino acid in N-terminal, and then here is a pathogenic reason, polyQ expansion reason, and following polychloride reason. This two part is very important because they are actually related to pathogenic mechanisms. PolyQ, and this is the reason form the pathogenic conformation, and then polyproline reason is actually affect pathogenic form of polyQ reason. So these both reasons are very important for Huntington disease. And I was able to crystallize this protein, which is N-terminal. I found the helical conformation, and the polyQ reason is loop structure, and then part of polyQ is formed the helical structure, and then another structure is completely formed helical structure. So this is show there is a pathogenic property comes from loop or helical structure of Huntington protein. And the Huntington protein pathogenic threshold is 36Q. So when you look at here, this protein is actually pathogenic length. When polyQ reason is 36Q, actually that start disease develop, and particular region of neurons are start to die. And so I thought if I crystallize pathogenic length of Huntington disease, maybe there is a particular structure will be actually related to pathogenic mechanism. When I solved the structure, it shows a helical structure, which is the same in non-pathogenic structure. And then there is a part of structure formed helix and loop. And then third form is I find that there is a partial of helical form and the hairpin form. And most people believe this helical form is related to pathogenic function of polycule region, but the answer is not confirmed. And another third crystallized ataxin 3, which is another polycule expansion disease. I crystallized this protein in 13Q, and I was very lucky because of, as you can see, every crystal, when I crystallize, there is 3 different conformations. So that is the difficulty of solving these proteins, because crystal has to be formed identical structure. That's how you can solve. But actually, all 3 of them are different conformations that give resolution is very low. It's about 4 angstrom resolution. But this one, I was very lucky. I was able to crystallize almost 2 angstrom resolution, which shows there. I see the polycule glutamine side chain is how they aligned along the helical structure. So actually that glutamine residue side chains are interacting each other and stabilize helical conformation. So once they, even if they are unstructured and can adopt different conformation, once they stabilize helical form, they can stabilize through the side chain interaction. So they can form very stable helical conformation. From these studies, I confirmed that polycule region of these proteins can adopt alpha helical conformation, loop conformation, and hairpin conformation, which is defined in crystal structure. And all these proteins are actually, when they are in solvent region, they actually inert. Inert means that they don't really interact with the other protein. They rather stabilize themselves in the solvent. So that is a little bit different observation. People think this polycule region is interacting many other proteins. But what our crystal structure shows is they are actually more inert. They like to be themselves in a solvent and stabilize their conformation. So polycule is able to form very stable helical structure by intramolecular interaction. Those are I observed from this crystal structure of a polycule protein. This is the most interesting part of this talk about polycule disease. I show here 8 different proteins, which is all same, able, when the polycule region is expanded, they have a toxic effect and kill the specific region of neurons. So, and then I look at these proteins, how they are different in physical characteristic. When you look at the amino acid side, their protein sides are very variant. They can have atacsin 3, they are about 360, compared to the Huntington protein is 3,000 amino acids. So protein size are very, very good. And then I look at the where is polyQ is located. Some are that located in amino terminal, and some are that located C terminal. And interesting part is this protein can have a very N terminal, which is below the 20, and then below the 50, and more than 100 amino acid reason. And then when I look at how these proteins are folded, the Huntington is a well-folded protein, so this ordered ratio is very low. Compared to this protein, ataxin 1, ataxin 2 is very unfolded protein, which is disordered region is very high. One thing interesting is this is a pathogenic threshold. Huntington has 36, 35, 36 region, and compared to the longest one is ataxin 3. Ataxin 3 has 54 polyQ expansion is required to pathogenic function. So when I look at this amino acid region, I group different three groups, and G1, G2, and G3. And this G1, G2, G3 is located from what is the end of a protein. For example, Huntington is from the 17 amino acid, but ataxin 3 is from the C terminal, but C terminal end from 46 amino acid. So depend on position from N terminal, which either N terminal or C terminal, how far from the end to the polyQ expansion. And then I think when these groups are different, how they can affect in proteasome function. If it is G1 group, they are more or less affected very early on processing proteasome, and then G2 group will be the alpha unit of proteasome, and the G2 group will be beta region of proteasome interaction. So G1 group and G2 group, these two groups are still ubiquitin related function, so they will be ATP dependent. Compared to G3 group is purely beta unit digestion, so they are different characteristic interaction with the proteasome. When in the very beginning, when I look at the polyQ structure, I mentioned about polyproline region, which is nearby polyQ region. And here we see there are three groups, G1 group, G2 group, and G3 group. But inside the group, they are separate again. Pathogenic threshold is different. How this pathogenic threshold is different is defined by this neighboring region. For example, early when I show the polyQ region, there is a Huntington polyQ region and polyproline region. There is a flanking domain. This flanking domain actually affect on toxic formation of polyQ. So when the polarity of flanking region polarity is high, then they destabilize toxic formation of polycule, so they require more longer of polycule expansion. And when polycule franking region is hydrophobic with low polarity, actually short region can form toxic conformation. From this study, I conclude location of polycule track in the protein sequence determine the different part of proteasome interaction and depend on early interaction with alpha region, alpha chamber and beta chamber. Their toxic mechanism is slightly different and they change the pathogenic threshold. Interaction between polycule protein and different parts of proteasome are affected by polarity of franking domain, which is nearby polycule region. High polarity increase pathogenic threshold, and the low polarity decrease pathogenic threshold. Second part is Alzheimer's disease, and the genetics already find out there are genes which is related to developing Alzheimer's disease. And there are two groups are divided by these genes. One is very high possibility for the developing Alzheimer's, and then there is a low probability of proteins, especially FOE and are the late onset, about 17, 18 years old, late onset Alzheimer's disease compared to penicillin and APP, and develop early onset, like earliest can be 40. So I am focusing on how these proteins are related to Alzheimer's disease. Penicillin-1 and membrane proteolysis protein, and APP is substrate of this penicillin protein. So penicillin cut APP inside the membrane and then generate A-beta peptide. And previous I discussed about A-beta peptide 40-42. So penicillin cut the APP and generate A-beta 40 and 42. And depend on 40 and 42, this pathogenic mechanism can be different. So my approach, I do not, as from the beginning I say I have a physics background. So I do not have a real experience with cell biology or neurons. So my approach is, already I have experience with the protein structure and mutation, how they affect the protein structure. And I try to use my strengths to understand physical mechanism of this APP-PS complex, how they cut the penicillin can cut APP in intramembrane domain. And for that, I use cryo-EM structure. Nowadays, many researchers work on large complex of proteins, and they publish and deposit. And I'm able to use these structures from database. And then using this structure, there are always information what kind of mutation actually can develop Alzheimer's disease. I use this mutation to predict what kind of mechanism involved in this process. This is penicillin protein, and I modeled the APP substrate inside in complex, and to cut APP by two enzymatic side, they has to have make a specific interaction. And I find that there are two kinds of mutation in penicillin. When you look at the penicillin, there are the 9 transmembrane domain. Actually, APP interacting transmembrane part is membrane 6 and membrane 7. And I look at the mutation in membrane 7 and membrane 6. And when I look, this is 3 helical conformation is interacting. So helical conformation to enzymatic activity is to cut the backbone. You have to stretch it, substrate. So to stretch substrate, they require certain energy to stretch this conformation. So I look at the which movement is dominant, whether transmembrane domain movement is or perimembrane domain is. So membrane 7 interact with the APP, mostly transmembrane region, and membrane 6 mostly interact with the perimembrane region. And I look at these two different mutation. When mutation happen, how this movement can be changed. Membrane 6 movement is actually reduced. Membrane 7 movement is increased. So membrane 6 movement is enhanced A-beta 40 generation. Membrane 7 movement is increased intramembrane activity. When I look, then if that is important in membrane interaction, how about APP case? When I look at the APP interaction where transmembrane interaction is making, there are mutations. When I look at the mutation, they also shows when the mutations happen in APP, actually their movement is increased. So it shows when intramembrane movement is increased, they increase A-beta 42 generation, and perimembrane movement is increased, then A-beta 40 generation happen. Then when this movement is where is coming from? Because this is a membrane protein, we can see penicillin can have two kind of movement. One is transmembrane. This is the transmembrane region. There are thermodynamic transmembrane region movement. Another one is where they interact is perimembrane region. As previously said, transmembrane region generate A-beta 42, and perimembrane movement is generate A-beta 40. So when I think about, for example, late endosome and early endosome, late endosome complement is compared to early endosome, they are very small. So they indicating they are more dependent on transmembrane thermodynamic movement compared to early endosome. They has to bend the curvature. They indicating there are more perimembrane movement from the early endosome. This indicates A-beta 42 is more likely late endosome or flat membrane surface can be generated from, and compared to 40, early endosome where it's big membrane bending is happen, they will generate A-beta 40. From this study, APP form a complex with penicillin, two different characteristic conformation, which is M1 and M2. M1 is APP form contact with perimembrane domain, and M2 is APP form contact with the transmembrane domain. By this, aminogenic mutation is PS1 mutation or APP familial Alzheimer mutation increase N2 conformation. By that, they are more generating A-beta 42 peptide. Also, membrane curvature shows APP processing M1 is favored curved, more curved better, generate A-beta 40 because of their big movement in perimembrane. Compared to the APP processing M1 is favored by late endosome, which is or plasma membrane, which there is much less curvature in the membrane, perimembrane, and there are interaction with transmembrane domain. That interaction generating A-beta 42. I just discussed about the mutation which is located in transmembrane region, but there are mutation in APP perimembrane region also related to Alzheimer's disease, and I want to know what kind of effect coming from this mutation. And when I first look at this mutation, there are more residue which is soluble to non-soluble, which is increased membrane interaction. So I calculate membrane interaction energy depend on how much you charge it. It shows when these mutations are all increased perimembrane interaction. And then when I look at compared to with using this different energy generation, I calculate relative how much A-beta 40 can be generated compared to wild type. When you calculate the value, wild type is 1. All the mutation in perimembrane region, they show the association of A-beta 40 generation is reduced. And also, I look at the A-beta 40, 42, 44, different beta peptide lengths. When I look at the A-beta 40, their membrane association is very minimum. But with the length increase, their membrane association is increased. And when I, again, calculate the relative amount of release from peptide compared to wild type, when the length is longer, there is a beta 40. A beta peptide is, generation is actually soluble. A beta generation is decreased. From this data is supported by other study. This is a study which is analyze all penicillin mutation. We grouped, depend on structure, we grouped 3 different reasons. And when the value wild type compared 1, A-beta 40, mutations are all reduced A-beta generation. And compared to the A-beta 42 is almost the same or relatively increased. when I compare the A-beta 40-42 ratio, definitely there is when wild type A-beta 40-42 ratio is 10, these mutations are all shows decrease of the ratio. So it shows in soluble A-beta has a certain function, which is we do not know what kind of function has this A-beta. But our data agree with other studies shows definitely A-beta and PS mutation totally shows soluble A-beta generation decrease and compared to A-beta 42. From this study, I was made a model which is when membrane surface endocytosis happen, endosome mostly generate A-beta 42, and then which is recycled, A-beta 42 soluble resin is released. Meanwhile, A-beta 42 is membrane binding. They can process to the further late endosome, which is membrane binding state. After this study, all study shows A-beta peptide mutation is increased membrane association. So why this membrane? Any peptide or protein which is interact with membrane, they have a specific effect on membrane conformation. And I try to find similar peptide which is similar amino acid sequence and structure. Then I think this structure have similar property like A-beta, which is associated with membrane. And what I find is 3 peptide, which is very famous for the antibacterial medicine. Actually, these peptides are binding in bacteria and take away the membrane and then break bacteria and kill the bacteria. When I compare to perimembrane resin of A-beta, actually they have a very similar energetic interaction with the membrane, but compared to A-beta 42, A-beta has much less effect on membrane association. So it shows A-beta 42 is definitely binding membrane surface, and they will adopt certain conformation which is effect on membrane surface. When I calculate the binding energy with the mutation in A-beta peptide, wild type A-beta 40 has a low membrane association energy. When they adopted mutated, their association is increased, which is more close to this peptide for antibacterial peptide. From this model, I can visualize that A-beta 40 is more generate soluble. They are endosome, they sort out soluble exocytosis compared to A-beta 42 generation will bend the membrane surface and remain with the endosome and process to the late endosome process. From this study present soluble A-beta peptide in endosome lumen might be essential for the synaptic and neural function, which we do not know what kind of function it has. A-beta 42 and longer A-beta species is more likely remain with the endosome, and then because of that soluble A-beta production, it will be reduced. the familial I2 mutation in APP increase association of A-beta 40 with the membrane, and that also decrease A-beta, soluble A-beta in endosome. Penicillin mutation shows that this mutation leads significant reduction in both total level of A-beta production, especially A-beta 40 is reduced. A-beta 42 and non-pathogenic non-aminogenic A-beta have, the conclusion is my study shows A-beta 40 has a particular soluble, A-beta 40 has a particular function, which is we have to develop it, we have to study about. A-beta 42 is enhanced endosome, late endosome generation, and that late A-beta 42 has a membrane association property like this, the bacteria antibiotic or peptide, so they able to form late endosome conformation. And from this study, I like to further investigate how A-beta 40 and soluble peptides is important with Alzheimer's disease. Another part is whether this A-beta peptide is actually related to ApoE function. So the end APP process, A-beta process in late onset of ApoE, how they are mechanically connected to each other. That is the final goal of my study. Thank you very much.